Assalamu alaikum. I am Dr. Khadiza from Bashundhara Ad-din Medical College. Today I am going to discuss about the erythropoiesis and its stages and the changes occurred in different stages. Okay, now we are going to discuss about what is erythropoiesis. Erythropoiesis means the process of formation and maturation of RBC under normal physiological condition is known as erythropoiesis. Now we are going to discuss the site where the erythropoiesis occurs. In fetal life, that is embryonic life, first trimester, that is first 3 months from the yolk sac, that is mesobalastic erythropoiesis occurs here, and second trimester, that is 4th to 6th month, it occurs in liver, spleen, lymph node, that is hepatic erythropoiesis, and the third trimester from 7 to 10 month, that occurs in red bone marrow and all bone marrows, that is myeloid erythropoiesis. Then extraembryonic life after the birth up to age 5 to 6 years, red bone marrow of all bones erythropoiesis occurs. Then 6 to 20 years, the red bone marrow of the long bone and the membranous bone, that is sternum, rib and vertebra and iliac fossa. This also vertebra, all these cases, all these bones erythropoiesis occurs. And after 20 years, only the membranous bones and end of the long bones are responsible for erythropoiesis. You can see this figure showing different areas of erythropoiesis in different years, that is 10, 30, 50 and 70. After you can see after 50 only the flat bones, that is sternum and vertebra, this is the area where the erythropoiesis occurs. Now we are going to discuss the stages of erythropoiesis. Firstly, we have to know some terms. Pluripotent cells, the cell capable of dividing and differentiating into any and all types of cells and even a whole organism. And pluripotent cells are the cell capable of dividing and differentiating into any type of cells but not to a whole organism. And multipotent cells are the cell with the capacity to differentiate into any type of cell in a given organ. And unipotent cells are the cell with the capacity to differentiate into only one type of cell. So this figure you can see the stages of erythropoiesis with the figures. First stage is proerythroblast stage, then basophilic erythroblast, then polychromatophil erythroblast, then orthochromatic erythroblast, reticulocyte stage, and then erythrocyte or mature RBC. Now we are going to discuss about what are the changes occurs all these stages in erythropoiesis. Firstly, the cell sizes decreases, finally nucleus disappears and the chromatin condenses and the nucleus become pycnotic and finally extruded. Cytoplasm become less basophilic and mitochondria, reticular materials and vesicles disappears and then hemoglobin accumulates. This is very important, you have to memorize all these changes in erythropoiesis. Firstly, we will discuss the proerythroblast stage. This is the first stage. In here, diameter is about 15 to 20 micron, and cytoplasm is scanty and basophilic, and nucleus is large with fine chromatin that contains several nuclei. Hemoglobin is absent here, and cell divide into by mitosis. The second stage is the basophilic erythroblast or early normoblast. In here, diameter decreases, that is 12 to 17 micron, and the cytoplasm is still scanty and more basophilic. Nucleus is large and the chromatin strands of the nucleus are thick and nucleoli have disappeared, and hemoglobin that is begins to appear here, and then here cell division also in mitosis. Now the stage 3, polychromatophil erythroblast or intermediate erythroblast, that is its diameter is 10 to 14 micron, and cytoplasm is polychromatic, that is bluish in color due to contain mixture of basophilic materials, that is RNA, and eosinophilic material, that is hemoglobin. Nucleus is small here and shows further condensation of chromatin, and hemoglobin its amount increases and cell division here also present. Now the stage 4, that is orthochromatic erythroblast or late normoblast. Here diameter is 7 to 10 micrometer, and cytoplasm is reddish due to a presence of hemoglobin, nucleus smaller, and condensed chromatin give a cartwheel appearance, and finally it undergoes pyknotic degeneration, and hemoglobin is large. Amount of hemoglobin is here formed, and cell division stops in this stage. Stage 5, that is reticulocyte. In here diameter is about 7 micrometer, and cytoplasm is contained network of reticulum, and the amount of reticulum decreases with cell maturity. Nucleus absent here, and hemoglobin fills the whole cell, and its concentration is about 34%. Stage 6, mature erythrocyte. Its diameter is about 6.3 to 8.8 micron, and cytoplasm is eosinophilic, nucleus absent here, and hemoglobin 3.34 gram per dl, and it become biconcave shape. So all these needs, and 7 days required for development of mature RBC from a proerythroblast. So from proerythroblast, 5 days required for reticulocyte formation, and another 2 days for mature RBC. Now we are going to discuss the factors controlling erythropoiesis. Firstly, the dietary factor. Protein is of high biological value. Then metals, that is iron, copper, zinc, cobalt, magnesium, and erythropoietin, that is a hormone, glycoprotein. And the factors that regulate erythropoiesis are hypoxia, vasoconstriction, nucleotides, hemolytic products, hormone, testosterone, adrenocorticotropic hormone, and thyroid. The maturation factors are vitamin B12, folic acid, vitamin B6, intrinsic factor of Castle, and vitamin C, etc. Now, what is the erythropoietin? It is usually a glycoprotein. Molecular weight is about 34,000, and its source is in fetal life from the liver, and in adult 90% from the kidney and 10% from the liver. Stimulation for formation of these hormones are hypoxia, vasoconstriction, nucleotides, hemolytic products, hormones, testosterone, adrenocorticotropic hormone, and thyroid. Usually hypoxic condition, when oxygen deficiency occurs, kidney secrete erythropoietin, which stimulates bone marrow for formation of RBC. This is the figure, shows how erythropoietin is released from the kidney and how it stimulates erythropoiesis. Now we are going to discuss the maturation factors. Usually vitamin B12 and folic acid is important for final maturation of RBC, and that is required for DNA synthesis. And how this is important, you can see when the deficiency of occurs of this vitamin, it causes defective DNA synthesis, and then failure of nuclear maturation and cell division, and the formation of large cells, then normal, that is normally called macrocyte of the cell, that is irregular and oval shape instead of biconcave shape, and then so this deficiency causes megaloblastic anemia, that is large RBC. Now intrinsic factor of Castle, you can know this factor is essential for absorption of vitamin B12 from the intestine, and deficiency of these factors causes pernicious anemia. And vitamin C, this is influence the folic acid metabolism. So these are the maturation factors and the how they cause. This table shows the different stages with important events. You can see early normoblast stage is the stage where hemoglobin starts to appear, and late normoblast stage where nucleus disappears, and reticulocyte stage, reticulum formation occurs here, and mature RBC, mature RBC is in here, near reticulum disappears and cell become biconcave shape. This figure shows the different stages with the changes within different stages. So that's all from today. Thank you. for watching.